is going to get unloaded to the tissues, right? So you can think of a scenario where you're exercising, as the case that I gave you. In that case, your hydrogen ion concentrations will start to increase through aerobic respiration. The oxygen is not going to get in as quickly as the need for making the energy. So your hydrogen ions will start to build. And so will your carbon dioxide, I mean exhaling it out. I showed you that little picture where they go hand in hand, that's the Bohr effect. So when you have that, the T state is going to be stabilized, more oxygen is going to be released to the tissues, right? So this goes back to one of the slides that I just showed you, where you have from I think 7.6 to all the way to 6.8 pH, and the graph shifts all the way to the right. So in this case here, here's my pH of 7.6. The pink one would be 7.4. And the yellow would be 7.2. Right? Okay, the shift is more towards the right as the pH begins to decrease, which means your hydrogen ion concentrations are increasing. Okay, so you're going to have to toggle back and forth with your pH. You're saying that low pH means high hydrogen ion concentration. So if you look at the questions, you can eliminate A. Right, so X is actually 7.6. Z is actually 7.2, so you can actually eliminate A and B. And then you can also eliminate E, because remember I just told you that pH has no effect, and it does. Right? And that's the Bohr effect. So the Bohr effect has to do with the pH and the carbon dioxide. So you can eliminate that. From here you can say that D is the correct answer, right? But let's go back to number C. That actually has to do with looking at your P50 and your oxygen loading, and things have to make sense. So with number C, as you shift more towards your right, the T state is getting stabilized. Okay, so we'll higher the hydrogen ion concentration, and you can go back and look at it. Go back and look at it and say that you have a hydrogen ion concentration, so P state is stabilized. And so you can go back and look at your slides where I summarized all this with the 3 different allosteric effectors and which state is being stabilized. And I told you to keep that close to you as you're studying and making sure that you're understanding why each one of them is getting stabilized, the T or the R. So as you push more towards your right, you're releasing more oxygen to the tissues. So that means the T, the deoxy state. So T is the deoxy state. That's going to get stabilized as you're pushing more towards the right. Does that make sense? Yes?
What's that in front of H?
Hydrogen ion concentration. That's a positive sign.
What's in front of it?
High.
Oh, okay.
Okay, high hydrogen ion concentration. All right. Does that make sense? Okay, so that's how you work on that one. Do you guys have any questions about this? You've got to know how to do these types of problems. Okay. Let's go back. All right, so we're going to look at the P50. The P50 P50 of X and Z. Because oxygen is getting unloaded more in the Z, you would, for question C, you would say that Z would have a higher number than X. If you don't see that, and you just can't do it and look at it. You have to make sure that the numbers make sense. Okay, so this is what you're looking at. So the number 2, we're asking for the P50, which is the pressure, the partial pressure, at which that hemoglobin is 50% saturated with oxygen. So on your y-axis is saturation, and so with X and Z, they are 100% or 1. So with X, we have 100% saturation, so we can call that 1, minus at a 50% range. Right? So with X, we've got about 20 torr. Okay? So that's the range, 20 torr for X. So because there is more oxygen being unloaded, the P50, the affinity for that oxygen, is supposed to be decreasing. Right? It's not going to hold on tightly. It's going to decrease, which means the P50 is going to increase with Z. So the higher the P50, low affinity of oxygen or hemoglobin, I told you that. But you've got to understand what that means. Don't just flatly memorize this. Because if you do that, you haven't understood it, you're not going to get a question where I actually ask you if you understood this concept or not. So as you're going more towards your right here, moving more towards your right, oxygen is getting more unloaded, so the affinity for that hemoglobin for oxygen is less. So P50 has to be on the rise. And that's exactly what you see. Z, right at 50%, is 50 torr. Sorry, 30 torr. 30 torr. Right? Okay. Good? Okay, so there's your 30 for that one, here's your 50. So that's P50. So definitely that question will be on the exam. Okay, so make sure you know how to do that. So that would probably be the simple question because you know how to do it. Or at least 66% of people in the class know how to do it. But, and you 66% know why it is the way it is. Right? So you should be able to get the question where I actually see if you understood what's happening. What's the relationship between affinity? What's the relationship between affinity and P50? What's the relationship between P50 and 2,3-BPG, like an altitude? Right? So those are things that you need to understand. You'll get this question right if you do it correctly and you bring your things down. That's why I say go over your worksheets again. A lot of people will do it here and then they don't look at it and then that's it, it's gone. But this should not disappear. Okay, so let's look at the last question. So we know that as you shift more towards your right, that's Z, the number for Z should be higher in terms of unloading compared to X, right? More oxygen should be unloaded with Z than with X because your T state is stabilized more in Z than X. Okay, more stabilization in Z than X. Does that make sense? Of the T. So your number should be that Z should be higher than X. If you get the opposite, then you've done something wrong. All right, so let's look at X first. So again, the numbers you need to know. Torr, 20 torr. Right? 20 to 30 torr. You just use 20 torr. It's just easier that way. But it can range if you want to look you're looking at. And then you look at the oxygen unloading. So that's the difference between the amount that's saturated and how much oxygen is getting released to the tissues. Alright, so picks the oxygen to the lungs, delivers it to the tissue. What's that difference? So that difference would be this height right there. So if we look at Z first, or X first, let's look at X first. So 1 minus whatever this range is, right, so right there. Let's say that's about 45.45. And so that'll give me 0.55, 55% oxygen unloaded. Yes? Question on that one? Okay, so let's look at the Z one, and you know the number has to be higher than 55%. More oxygen, we just stated that T is stabilized, and more oxygen has to be unloaded. Alright, so now we look at C, so it's the difference here, right, same thing, it's 100%. So 1 minus my Z here, which is up to this point, right there. That's my Z. So 1 minus 0.25, which is 0.75, which means 75% oxygen unloaded. Good, yes?
Why does a higher pH mean a lower affinity?
The pH, the hydrogen ion concentration increases. And so if we go back to Y, the hydrogen ion concentration is going to couple to the histidine group of the T state, which gets exposed and stabilizes the T state. So that's the how. So you gotta know the how is doing it. And you gotta know what's gonna be the relevant consequence of it. Those are the two things you gotta think about. The how are things working, the mechanism, and the functional consequence. Did you have a question?
So on the previous question, we said that at P50, torr was 20.
Let's see, the X is 20, the Z is 30.
Yeah, so when we're doing question number 3,
Yeah.
can we look at the tissue at 20?
Yeah, well, no, I'm just looking at
Just an estimate?
estimate of that. So the P50 it can vary, right? It can vary depending on what the protein is. If your graph looks like this, you're gonna have to use that saturation of 0.6, not 1. Right? You gotta look at your graph and see where it's plateauing, where it's getting saturated, that molecule. So it might not necessarily be at 1. It might just be at 0.6. And from there, again, you do a half, 0.3, and then you bring it down. Does that answer your question? Yeah? Yeah.
We use 20 torr in this calculation because that's the pressure.
That's the pressure at which that hemoglobin is going to be bound to 50% of the oxygen. That's the pressure. That's what it means. Any other questions? Yes?
So even though like on the slides and stuff, the tissues was at 20 and then the ones were at 100, that doesn't like matter or anything?
No, those are two different things. The pressure of that oxygen that's fully saturated in the lungs is 100. That's the measurement that's done. At that 100 torr, all of that hemoglobin, because that's physiological, so you're looking at that range, and that's going to be the pressure at which all of that hemoglobin is saturated. And then at the 20, when it's coming to the tissues, the pressure is at 20 torr, and so you're releasing that. So the difference between those two is your oxygen unloading. So that's different from the pressure at which that hemoglobin is 50% saturated. So those are two different things. But they do go hand in hand, right? If you're the higher the P50, the lower the affinity for that oxygen. But at the same time, the higher the P50, the higher the number for oxygen unloading will be. Right? That's what you saw there, because remember the Z was 30 torr, higher than the X value of 20 torr, and you see that same correlation. Right? Z is also higher. So higher the P50, that means you're unloading more oxygen. So both those numbers have to be higher than the one that's not unloading. Good? those two do go hand in hand. Anything else about this?
Could you possibly like post another example, but like not where it's at one, like where it doesn't plateau at one?
Yeah, I mean, you can make this, let's just do this now, let's just make this at 6 here, 0.6. Okay, my graph is not great, so here's your 0.6. And then I just go at 0.3. Right? And so that'll be at 30, so my 0.6, I do half of that, it's at 0.3, and my P50 at that state would be, what, 30?
Okay, yeah.
That's all it is.
Okay, that makes sense. Thank you.
Because you're doing a half.
Yes,
That's all you're doing, you're doing a half. So make sure you're looking at where that saturation is happening.
Can you do now for 20, like you do 20 for the tissues, right, to find the other value?
For the one that we just did, this plateau one,
Yes, ma'am.
okay, so it's plateauing here, same thing. Let me use another color. Can you still see this color?
Yes, ma'am.
Okay, so there's your 100, and then here's your 20. And so the difference, right? So the oxygen unloaded is 0.6 minus, where is it? I'm losing track here. About here, to 0.1, right? 0.5. So about 50% is unloaded.
Okay.
That's the difference we're looking at.
Thank you.
All right, so kind of walk through it with yourself, do the examples from the textbook. There's a couple of examples you can do from the textbook, and I'm throwing in the assignment as well. So take a look at that. And if you have any questions, you can always email me or come by office hours on Monday. I'll be here. All right, so let's go to our second biomolecule, which is carbohydrates. Okay, so we just started with proteins, your amino acids, obviously, but we're not quite done with proteins. We'll come back to it again with molecular biology halfway through the course, but the amino acids will continue to come even today. So now we're moving on to the second biomolecule, which is carbohydrates. So I'm going to tell you, I think most of the things are underlined here. So anything that's underlined there, you need to know. Okay? And very simple. You won't need me for this because you just have to memorize this. Find a way to memorize it. Talk it with your friends. Do whatever you need to do. Make a table. Just memorize them. All right? So we start out with the simplest carbohydrates first. And so I'm going to tell you which ones we need to know because those are the ones we're going to use this semester, and for those of you who are planning to take 322, those are the ones we're going to continue to use next semester, so you need to know them now. Common disaccharides, there'll be 3 that you need to know. We're going to talk about polysaccharides, the features of those polysaccharides, that's structural features, their storage molecules, but you need to know their features, right? Look at the structure of them, and then the biological functions of them. We're going to look at glycoproteins, proteoglycans, mucins, lectins, all different types of proteins that have carbohydrate groups attached to them. Okay, so these are proteins with carbohydrates. So that process of adding is post-translational. So the protein is made and then it goes into the Golgi, and there something happens. In this particular case, we're looking at adding carbohydrates to your protein. Okay, so that's really what's called the topic of glycosylation. That's adding your carbohydrates to your protein. Yes?
What did you say we needed to memorize?
I'm going to tell you.
Okay.
Yeah, I'm just giving you what's coming up, but I'm going to tell you. As far as like more of the clinical insights, there's several that are very interesting. I'm throwing in penicillin because it really has to do with a lot of the carbohydrate structures on the lipid, the bilayer, peptidoglycan bilayer. So make sure you read these 2 pages if you need a little bit more information than outside of this lecture. And then Tamiflu. How is Tamiflu affecting and stopping viral progression? So lots of different functions of carbohydrates. We're going to talk about some of them today. Really the energy is probably the main thing. And as far as energy, we're going to talk more so in 322 when we actually metabolize glucose. We have today talk about the cell components. Obviously, these membranes will have carbohydrate groups attached to protein, so they are part of the cell membrane process. They are a stored form of chemical energy, because when we talk about, for example, glycogen, which is a polysaccharide, it is groups of glucose attached together, and it's stored. Because remember, we don't want all these glucose molecules in our bloodstream and have a high glucose level. So the liver will store them in the form of glycogen. Okay, so that's where that stored chemical energy comes from. And of course, the structures of some cells and tissues will have different carbohydrates. So these are some of the examples that we're going to look at. Monosaccharide means one sugar. Disaccharide means two sugars. Oligosaccharides are fewer than 10, more than 2 but fewer than 10. And then polysaccharides, we have more than 10 groups of sugars attached. And so these are some of the examples. We're going to look at monosaccharides, we're going to look at disaccharides, and we're going to look at polysaccharides. I'm not going to really look at the oligosaccharides, except for how attachment occurs. Okay, how do these sugar groups attach, and what's the enzyme that does that work? So when you classify sugars, carbohydrates, they're either going to be in the aldehyde group or the ketone group, two forms. Okay? And again, CHO. So in the beginning of chapter 1, I said that carbon, hydrogen, and oxygen are going to be part of all biomolecules, and that is the case for carbohydrates too. You'll see the carbon, hydrogen, and oxygen.
Can you go back real quick?
Yeah.
So it shows
Yeah, everything is on your PowerPoint slides on Canvas, so you don't need to copy these things down. Take the notes that you need to take as you feel fit, okay? You don't need to copy these things down. All right, so let's look at the monosaccharides, aldehydes, or ketones. So two groups, so that's how you classify them. And so when we look at these groups here, we have simple sugars. They're simple sugars because there are 3 carbons. We're going from 3 carbon all the way to 6 carbon sugars. So these are the simplest form. So there's 3 carbons. See, 1, 2, 3. And this dihydroxyacetone is an isomer of the glyceraldehyde. So dihydroxyacetone is a ketone group, right? This is your ketone, right? And there's your aldehyde. So that's why they say it's a ketones family or the aldose family. And they are isomers of each other, and that's shown here. Okay, so here's your glyceraldehyde and your dihydroxyacetone. They are isomers of each other, which just means that they have the same molecular formula, just rearrangement is different. This should be reviewed, hopefully, but go over this table if you need to, the definitions, so we'll go through them here. So isomers, like I just told you, have different molecular structures, different molecular formula, but different structures. That's the example that I just showed you. See, the C3H6O3 is the same also in the dihydroxyacetone, so they are isomers of each other. Stereoisomers means that the atoms are connected in the same order, but are different in spatial arrangement. You can have an enantiomer, which is non-superimposable mirror images. They can be the D form or the L form, or they can be diastereomers, which are isomers that are not mirror images. Those diastereomers can be divided up into 2. Epimers and anomers. When you look at the anomers, you see where they're different in the asymmetric carbon and how that OH is grouped up or down, right? And then when you look at your epimers alone, they differ at one of those several asymmetric carbon atoms, and I have starred where that asymmetric carbon atom is. So you need to be able to classify them, whether they belong to the aldose family or the ketose family. Okay, so anything with an aldehyde is an aldose, and anything with a ketose is a ketone. Anything with a ketone group is a ketone. It should say ketone. Change that to an N, so it should say ketone. Then you have to classify them based on numbers, and I'll tell you which sugars you need to know. A 3-carbon sugar, like the one that I just showed you, like a glyceraldehyde, would be a triose. That's 3 carbons. Anything with a 4-carbon would be a tetrose, a 5-carbon, a pentose. Glucose, that we're going to look at today as well, is a 6-carbon, and it's a hexose. So your book, I think, has something similar to this, but I really like the first edition version where they divided up the aldose and the ketose family this way. So the aldose family, the ones that are underlined are the ones you need to know. Not a lot, there's about 5 here. Glyceraldehyde. This is all aldose family. Glyceraldehyde is one that belongs to the aldose. It is 3 carbons. See the 3 carbons there? It's already written down on the slide, 3 carbons, so it's a triose. So if I give you glyceraldehyde as a molecule and I ask you to classify them, you should be picking out aldotriose. The next one that I need you to know is ribose. Ribose will come in the DNA and RNA structure. It is a 5-carbon sugar. That's why it's a pentose, and it belongs to the aldose family. Glucose, mannose, and galactose are also important here. We'll be looking more specifically at galactose next semester and how it can form cataracts. But all of these ones, these 3, all belong to the hexose family because there's 6 carbons and they are the aldose family. Same thing here with the ketone group that belong to the ketose family. I need you to know 2. Dihydroxyacetone. Remember, dihydroxyacetone is an isomer of glyceraldehyde that I just showed you. And the fructose also is one that I need you to know. It's very important especially for 322, but you need to know this one. It belongs to the hexose families and ketone group. So it's a ketohexose. Okay, so simple memorization there. So when you have aldehydes, they can react with alcohols just like sugars can. And I'll show you the straightforward structure in a minute for sugars, but remember from organic, the aldehyde can react with an alcohol to either create a hemiacetal, or if you have a ketone group and the alcohol group reacts with it to create a hemiketal. So you can think of this in terms of sugars, and when you look at a straight chain carbon like glucose, an open chain like this, you're doing the same form of a reaction with an alcohol with an aldehyde group. And so here's your alcohol group, this is your chain here in the ring form, which becomes into the ring form. Most of the glucose molecules or any sugar likes to exist in the ring form. So the alcohol will attack the carbon 1 of the aldehyde group to create that ring form. And it can be in the alpha or beta structure. So they give the name glucopyranose, the pyranose, because the structure itself looks like a pyran, so that's where the name comes from. So that's a pyran group. All right, so you can do the same thing with a ketone group family, the ketose family, and the 6-carbon sugar shown here is your open chain form of fructose, and the alcohol can attack the carbon 2 of the ketone group and create fructopyranose, the furan family here. This is what it looks like, and that's where that name comes from, of fructose pyranose. Okay, so this all happens with 6-carbon sugars. Whether we're talking about aldose or ketose, you can create that hemiacetal or hemiketal group. In nature, it seems like more of it, when they look at it, we're really not sure why that's the case, but more of it, they like to be in this pyranose family group. Okay, if you look at the percentages wise, most of these sugar groups that are forming the cyclic ring will be in the pyranose family. You want it to be in this form here, the chair form, because of this steric hindrance with the boat, so it formally forms in the chair form. For those of you guys who are going on into the lab, BMD 323, you'll be doing a carbohydrate experiment where you're given different sugars and trying to classify them as whether they're reducing or not. What makes it reducing is having an exposed aldehyde group. That's the definition. Okay? So any carbohydrates that are aldehyde groups that are aldose family are considered to be reducing sugars. So you take the sugar, this is your copper, which gets reduced here, and you're going to oxidize the sugar, and when it gets oxidized, it changes color into a dark brick red. And so when you see that color change of a dark brick red, that tells you that this unknown sugar that you have in your hand is a reducing sugar. Okay, so anything that has that aldose family can be reduced. But remember that these sugars can isomerize. And in the case of the glyceraldehyde and dihydroxyacetone, they can isomerize, especially if you put it into the base. And when they do isomerize, you can actually convert something that's in the ketone family into an aldehyde family, and you can make it a reducing sugar. And in the lab, you'll get fructose, and you'll see that fructose, even though fructose is a ketone family, there's a base in the reagent, it converts, and you can make it into an aldehyde family, and that becomes automatically a reducing sugar. Okay, the color will change, and the question will ask you, why is fructose, even if it's in the ketones family, why is it reducing? So the answer would be it would isomerize. All right, so those are reducing sugars. Do you guys have any questions so far as to what you need to know? Yes?
What did you call fructose?
The fructose? The fructose isomerizes to glucose, is that what you're asking?
Yes.
Yeah, this is an isomerization reaction. So really when you're talking about isomers, they are the same molecular formula, right? Just rearrangement. So you can count up the carbons, the hydrogens, and the oxygens there, and you'll see they're the same. So that's what an isomer is.
So those are isomers?
Yeah, those are isomers. Okay, so any type of monosaccharides can be modified. I'm just going to point out to you really a couple of them here. Glucose. Glucose can be phosphorylated in the carbon 6 position to become a glucose 6-phosphate. That's actually the first reaction that happens in glycolysis. So there's one of your modifications. Same thing here. We're going to see dihydroxyacetone phosphate next semester in glycolysis. It is a sugar. Dihydroxyacetone is a sugar. That's one of the ones you need to know. And it gets phosphorylated. Okay, so this again it belongs to your ketone family, and it's a 3-carbon. All right, so that's one of the ones that I underlined for you that you need to know. But these two sugars you'll see next semester. Today we're going to talk about sialic acid because that has to do with, if we have time, we're going to talk about sialic acid, but that has to do with Tamiflu. Okay, so it is a modified sugar on the surface of our cells. So how does the modification, well, we're going to see some of these different other types of modifications. So we have a sugar here, here's your anomeric carbon 1. It can get modified, and when it gets modified at that carbon 1 that has that OH group, that bond that it's getting modified to, that bond is called an O-glycosidic bond. We can have an N-glycosidic bond, okay, so why an O? Because there's an oxygen bound to that modified group, so that's why it's called an O-glycosidic bond. We can also have an N-glycosidic bond as well. And when we have an N-glycosidic bond, when will you see that? You'll see that later in the semester with this nucleotide, which has a ribose attached to the base. Okay, so that's why it's because it's this sugar group. This is your sugar, right? This is one of the ones you need to know. This is a sugar attached to a base. Linkage is through the nitrogen, so it's called N-glycosidic bond. All right, O and N. Okay, so that's all monosaccharides. So how do we link them together? We're going to show you the reaction of the enzyme that's used, and disaccharides link 2 sugars together. Oligosaccharides are two or more sugars together that are linked. Okay, so where is the linkage happening? So here we have a linkage here of a maltose, and that sugar group, two sugar groups are attached together from malt, and you can actually digest maltose from the enzyme maltase, and you have your O-glycosidic bond right there. So those two can bind together because there's an alcohol group and an alcohol group here, releasing the water, creating that O-glycosidic bond. So this bond is an alpha 1,4. Why called an alpha 1,4? Because the link is between the carbon 1 and the carbon 4. Alright, so that's where the name comes from, alpha 1,4 glycosidic bond. So you've got O-glycosidic bond and you've got N-glycosidic bond as well. So these common disaccharides you need to know, all three of them. So you need to know which two sugars make up the saccharose, the lactose, and the maltose. Alright, so lactose, let's start with lactose. Lactose is milk sugar. So that one is a combination of galactose plus glucose. I don't need you to know if it's a beta or an alpha. They're all O-glycosidic bond, right? Any disaccharides that you build together are going to be O-glycosidic bonds. And you see that O-glycosodic bond here, an O-glycosidic bond here, and an O-glycosidic bond there. Right? All three are O-glycosidic bond. And it doesn't matter what disaccharide it is. They're all going to be O-glycosidic bond. That's how they're going to join. Sucrose, you can break. That's your sugar from. You can see that in your sugar. You put your milk, coffee, and then you put that into glucose and fructose. Okay. Maltose is glucose plus glucose. Two glucose joined together. This is different from mannose, okay? So don't confuse the maltose with the mannose. So that's what you need to know, the combination. So easy, 1-point question. So those are the common, really, disaccharides. And we will actually be looking more specifically at lactose digestion. So that's why it's important for you guys to know the combinations of these. So when we join these sugars together, we do have an enzyme, different enzymes for different sugars that are being bound together. And these enzymes are called glycosyl transferases. Transferases, right? You can tell just by the word of it, it's going to transfer from sugar group to another. So to build things, right, to attach things together, central theme is you need to put energy into the system to build things. When you're breaking things down, you're going to generate energy. So since we're building this together, we have to take the sugar, here's your sugar here, and couple it with a high energy molecule called UDP. This is somewhat similar to an ATP, except there's a uracil. And we really don't know in nature why some specific reactions use UTP, some use ATP. Okay, in this particular reaction, we're using UDP that's coupled to the sugar. And when it's coupled to the sugar, the enzyme transferase is going to transfer this sugar to another sugar X. So any sugar, and you create that linkage. And that linkage again is going to be your O-glycosidic bond. Right? Between the two O's, between the O. Okay, so that's how you activate it. You always have to put some energy into it. That'll really be a theme, central theme within Biochem 2. Do you guys have any questions about what you need to know about disaccharides? All right, let's go move on to polysaccharides. The one in the stars are ones you need to know that we're going to at least talk about today. Structure-wise, you need to know the structure of glycogen, starch, cellulose. All right, and what they do. Glycogen and starch is storage. It's for storing those glucose molecules. In structural form, the cellulose is present in plants. It's for structural purposes. So the structure itself will be a little different between the cellulose and the starch, both in plants. And then we're going to also talk about the polysaccharides within the bacterial cell walls, and this is where we're going to talk about penicillin. Okay, that's where the relevance comes in. Penicillin. When the same sugars, say glucose, all our glucose molecules bound together, they're the same. So when they're the same, they're called a homopolymer. Okay, so polymers are chains of something. Polymers are chains. And since we're talking about carbohydrates, they're chains of sugar groups bound together. And when you have all glucose molecules bound together, they're homopolymers. They're the same, molecules bound together in chains. And that's the case for glycogen, starch, and cellulose. They're all glucose molecules bound together. And they're just being a different structure. So polysaccharides wise, glycogen versus, let's talk about glycogen versus starch first. Glycogen is in liver, right? So there's your glycogen in the liver. Right, that's our storage. So liver stores our glucose molecules. So like I said, you don't want a high level of blood glucose level in our bloodstream. When they don't need to be used up, then they're not used up, and it needs to be stored, right? So liver will go ahead and synthesize glycogen by, again, some of the enzymes that we're going to talk about, that join together those glucose molecules. When we talk about starch, we're talking about plants, like potatoes, right? Starch will have plants, so that's where we're going to talk about, but they're really structurally different. But in either case, you can digest them out with an enzyme called amylase, right? So you can digest glycogen or starch with an enzyme called amylase. Simple memorization. What does glycogen look like? Okay, all sugar molecules here. Okay, so here are all your sugar molecules, and those are all glucose, right? They're all glucose molecules. So with glycogen being a storage, all the glucose molecules are linked together. And the way that glycogen is built is you have O-glycosidic bonds, right? An array of glucose molecules bound together by O-glycosidic bonds. And every 10 or 12 groups ahead, you'll have branching to a 1,6 glycosidic bond. Okay, so this one has a branching. Glycogen has branching. It has a chain of alpha 1,4, and then 10 to 12 units away, you'll have a branch occurring at the 6th and 1 carbon to give you that alpha 1,6 linkage. Like I said, it's high in the liver. It synthesizes as it is in the muscle. You'll synthesize it and store it in the form of glycogen because, again, you want to maintain that blood glucose level. And you don't want that high blood glucose level, so you've got to be able to store it, and this is the form of storage. So that's why it's a function for storage. Starch, it's in plants. In plants, they come in two forms. They come in amylose and they come in amylopectin. So amylose is quite similar, the smaller name, perhaps you want to remember it that way, the smaller name, simple structure, all alpha 1,4. Okay, no branching. So all alpha 1,4. The longer name amylopectin is a little bit more complicated, but it looks very similar to glycogen because it's branched. So it has alpha 1,4. Instead of 10 to 12 units away, it's a little bit further. Every 30 units apart, you'll see a branching of an alpha 1,6. And all of this starch and glycogen can be degraded and broken down by amylase into disaccharides, and then you got to do another step, which you'll learn about next semester. All right, cellulose is not a storage. It is a structural. Like I've noted down here, it's a structural component, but it's in plants. And the difference between all of the ones that we talked about is, again, it is a homopolymer, just like the other ones. The main difference is it's connected by beta 1,4. Alright, I'm showing you that beta 1,4 linkage. Okay, so it may have 300 to 3,000 units of glucose bound together, but they're all glucose chained together, unbranched, just like amylose is unbranched, but it is an alpha, it's a beta 1,4 glycosidic bond. Okay, so all the notes are there for you guys that I just talked about. You just have to memorize it. When you look at cellulose and you look at starch and glycogen, this is sort of a hand waving. I don't know if this is really something, just looking at the structure, this is kind of how they rationalized why it's a beta 1,4 versus an alpha 1,4. When they looked at the structures, starch and glycogen being storage, right? They're storage molecules of glucose. They have more of a hollow shape. They figured that that structure itself impacts how the storage occurs. When you think about structural, the beta 1,4, look at how compact this is, and they rationalize that because they are packed like that, it's important for structural features. Do you guys have any questions about those polysaccharides, what you need to know? Okay, so let's advance a little bit further into the topic of glycobiology. So really, it's glycobiology, like the name says, it's the biology of carbohydrates. So we've got different types of proteins that have different types of attachments of carbohydrates. We're going to talk about glycoprotein, proteoglycans, and mucins. They're very specific types of carbohydrates that are attached. And you just need to memorize what type of carbohydrates are attached to these different types of proteins. So we talked about O-linked, right? And we talked about N-linked, how that occurs. Specific amino acids on the proteins can be O-linked or N-linked. That's the key, right? So that doesn't mean that every serine, in this case that I've given you here, serine here, every serine on the protein has a sugar group. It just means that what they've seen is serines have sugar groups on them. Okay, so wherever they look at a protein and a serine, they may have a sugar group on them. That doesn't mean all serines are going to have sugar groups on them. So you know that serine has an OH group. So serine would be one that would have a O-glycosidic bond. So here's your protein, right? And you know this is a serine. Here's your R group with an OH group to it. And that is linked to some specific sugar. You don't need to know the name of the sugar. But that protein has a serine. There's a possibility that a sugar is attached to it. And this is a form of post-translational modification. So throughout the semester, you've been learning about different types of post-translational modifications. That means the protein has been made and then gets modified by going to the Golgi. So in this case, you're having glycosylation, which I have put somewhere here on the bottom, right? So I put glycosylation as a term. So that's the form of post-translational modification. Modification of the protein after the protein has been made. So these proteins that have sugar groups attached to specific amino acids are called glycoproteins. Glycoproteins are the most highest majority that we have that make up our system, and that includes various roles like membrane proteins. So they can be O-linked or they can be N-linked. And so can you guys think of other areas where you can have an O-linked between a protein and a sugar group, other than serine? What else? Yeah, tyrosine. That would be another one. That would be, have an OH group to it, so you can read it. Okay? And they can also be N-linked as well. So here's your protein. Asparagine is the only one that they've seen so far that has that N-linked protein that through the nitrogen binds to a sugar. Not to be memorized or anything, but there is a pattern, okay? And this pattern is shown here. And this is really important. You're kind of wondering, why are we studying all these patterns and memorizing these? And if you're a person in the glycobiology field, this is very interesting. And let me come to, it's coming up later, I guess. Okay. And this is actually very important because they studied blood groups this way. And so I'll talk about the blood groups in a minute. Erythropoietin is another example of a protein that has sugar groups attached to it. And, proteins that have sugar groups attached to it, they do have a function. And so one of the functions of those sugar groups attached to a protein is stability. So they do actually stabilize protein structure. Okay? And so when athletes take erythropoietin, they're able to tell if it's actually the regular one that the body produces or something that they have taken. And that's basically by looking at the patterns of the sugar groups attached to it. There's a difference between the sugar groups that we normally make, erythropoietin, versus that is taken by the athlete. So they'll be able to tell if they've taken this particular molecule for endurance purposes. Proteoglycan is the next subdivision that I want to talk about. So it's a particular protein that has specific polysaccharide linkages. It is made up of 95% of the proteoglycan has this sugar group, glycosaminoglycan. Okay, so that's the name of the sugar. So there's more carbohydrates than there is protein. It's the reverse for glycoprotein. Glycoprotein, there's more protein than there is sugars attached to. So what are glycosaminoglycans? They are repeating units of disaccharides, and they are derivatives of an amino sugar and one that carries a negative charge. And that could either be a carboxylate or a sulfate group. So they do have a negative charge. And that's important because they are important for extracellular matrix. They serve as structural roles, like when you hit your foot, it rebounds because the water is able to hold on to that negative charges and it cushions it. Okay, so that's really the part of that extracellular matrix, giving you an example. So when you look at structure-wise, these are the two sugars. These are the glycosaminoglycan. All right? The glycosaminoglycan have these two sugars. Either of them, they can be two, they can be the same, they can be different. Plus, they have the carboxylate group. Okay, so they have the sulfate group or they have the carboxylate group. So the definition of these glycosaminoglycan is these two sugars with the carboxylate or the sulfate group. So you have the negative charge to these proteoglycans. So this was the example that I was telling you with cartilage, where you hit your foot, you rebound because the water gets absorbed by the negative charges on those proteoglycans. And these cartilages have these proteoglycans like aggregin, for instance. That's a proteoglycan. And the last one that I'm going to talk about is mucins, which is really a specific type of protein that's found in enriched in cancer patients because they're important. They are there for metastatic purpose. So one of the functions that we talked about was with sugar groups is stabilization. The next class we'll talk about how it communicates. With mucins, another mechanism is one is of course for you see the word mucins and you see protective barriers within the lungs. But if you have mucins and they are glycosylated, and they are, they're specifically O-glycosylated, they will migrate from one position to a distal organ. Okay, so there is that communication between cell types through the carbohydrates. So we've talked about communication, cell-to-cell communication, and we've talked about stabilization as far as having carbohydrates to the proteins. So we'll come back next week and continue on further, but if you guys want to look at this video of how sugar groups are attached to blood and what's going on with that.